Protein synthesis is one of the most fundamental parts of life. It's also one of the most difficult topics that you will be looking at this side of your GCSEs. It's something that every organism does. You, me, these trees, the bacteria on the floor around me. And it's why you have DNA, and it's why that DNA makes you, you. It's one of the first processes to evolve as well, and we know that because whether it's you, me, this holly tree over here, or the bacteria on the floor, it's exactly the same process done in exactly the same way. So in this video, the first of the week, we're going to go through the basics of the process, and we're going to start to understand how we go from the information in your DNA molecules to a useful, functioning 3D protein. But fundamentally, proteins all have one thing in common. They're all just a chain of small building blocks that we call amino acids. There are 20 amino acids that we find in nature, and the order that we put these in our chain determines whether you have keratin in your hair, hemoglobin in your blood, or actin in your muscles. And the instructions that tell your cells which amino acids to put in which order are your genes. So we need to understand how your cells can take the information in your genes and turn them into this functioning chain of amino acids. And for that, we need to head to Castle Lane. Now, this may not be the most impressive library you've ever seen, but it's an important one. Each of these books contains dozens, if not hundreds, of recipes. Instructions, if you like, to take a simple bunch of ingredients and turn them into something more complex. Each recipe then is like one gene, one set of instructions to make one protein. And the book, which contains them, is like one of your 46 chromosomes. Now, the simplest way of getting this information into the kitchen where I need it is just to take the book. But there's a problem. I've only got one copy of each book, and it's delicate and it's valuable. If this book goes, all the instructions go. That means I've got a job to do here before I go in there. So I have a copy, same information, same language, but a transportable and disposable version of it. This is a process we call transcription, and this is a molecule called mRNA, messenger RNA. It's the messenger molecule that will take the information from here in my library with my books into the kitchen. Now this is where the magic happens. In this analogy, the kitchen is the cytoplasm of my cell, and the chef is the ribosome. Now, the ribosome is going to take the information in the messenger RNA, the mRNA, and it's going to use that to build up a complex product from the right order of amino acids at the right place. So what I get at the end of this process is a protein. Now, I've taken the information, the genetic code in my mRNA, and I've used that to change the language now into a sequence of amino acids. And because I've changed the language, we call this process translation. So there you have it. That's the whole story. Now, okay, of course, it's not the whole story, and we've got a lot of nuts and bolts and little bits and pieces of protein synthesis to cover over the next few lessons. But it's a nice overview of transcription and translation. Awkward ending. I didn't plan this bit. Walk out of frame, good walk out of frame. But I don't like that. How about this one? If I straightforward cut to black. I'm not sure about the cut to black. Weird fall. How's that? Meet the dogs. Dog 1. Dog 2. Stars the show, really. You pick the ending you like best. Just cut back to the ending you like best.